Hi everyone, this practical experiment is about wet mount preparation. We need to prepare a microscopic slide to study cells. Now in our task we are preparing cheek cells and onion cells. Cheek cells is an example of animal cell, and onions, we're using onions as an example of plant cell. Now, for the materials that we need for wet mount slide preparation, I need you to memorize them because they're common in wet mount slide preparation. I need microscopic slide, cover slip, and from the name cover, it covers the sample. Now, why do I need to cover the sample? I need to protect my sample from air bubbles, because sometimes air bubbles are really annoying under the microscope, and I'm protecting my sample from being polluted with fungus or bacteria or whatever. So it's for protection. I need over here a dropper for the wet liquid I'm using to help me and to view the cell. The liquid I'm using is water, very simple, but with the water I will see the image colorless, without colors. And in order to see the image colored, I'm going to use a stain. Now I can use methylene blue, this one. I can use iodine, I can use methylene red. It's just a dye, like an ink, let's say. It only dyes the cells without affecting the cell, without affecting the nucleus, without nothing. It reacts with the nucleus and it makes the image clearer. So in order to define the stain, it is a dye, react with the nucleus, it makes the image clear. That's it. Okay? So in my case, I'm using methylene blue, not iodine, not methylene red. I love blue color. This is the reason. For cheek cell, I need wooden stick. For any other sample, I need forceps or tweezer. So mainly, these are the materials needed. To prepare the sample, very simple. I will add a drop of water and drop of stain on the same slide. Just not to waste my time doing many slides. I will just do it in one. So I will add one drop. You know, usually in the lab we are preparing everything over here, and we are using gloves, and we are using wearing a lab coat, of course, but I am doing my own, so I know what I do. So this is a drop of water, and this is the drop of stain. Now, how would I collect my cheek cells? I will simply scratch my inner cheek from inside, not the tongue, the inner cheek, okay? And once I'm done, I will apply the sample on the water and give it a little bit mix. Now, why do I have to mix it? Just to make sure that I'm collecting all the cells from the wooden stick into the drop of water. And I will, you know, like take another side of the wooden stick, or use another wooden stick to collect another sample for the drop of stain. I won't use the same spot over here. I will change and give it a little bit mix. Once I'm done, I will get rid of it. Now, when I apply the slide, I should tilt, keep it on the side over here. Can you see it? I should just tilt it, and very slowly, very slowly, very slowly, I will apply it. Now, why slowly? I want just to drop it, because if you drop it, you'll create air bubbles. Look over here. clear, no air bubbles. If sometimes you see 1 or 2 air bubbles, with the help of the tweezers, you can very gently, or pen, just tap, very simple, to remove the air bubble. But when you drop it, you tilt it and put it very slowly on top, you won't create any air bubbles. If you drop this cover slip, there will be tons of air bubbles that will really annoy you under the microscope. And the same for the, I'll just do it again. So I will tilt, see, I will touch the sample over here. See, this is the air bubbles I'm talking about. You will simply try to tap, follow it. Of course, I need to do it on the surface of the table. And this is cheek cell sample. Very simple. Now for cheek cell, same 2 drops, water and stain, but in this case, I need to do onion. Did I say cheek cell at the beginning? Yes, onion, real life onion. Now, why did I cut the onion? Because when I cut it, I need to peel this peeling over here, the outer peel. Can you see it? Very thin, or from inside. And this is better, much more easier. Look, this is the one I'm using. See? This is the one I'm using. So I will just apply it on the water. And I have another piece over here. See? This one. I will apply it on and I will add my cover slip as well. Again, I will tilt and try not to drop, very slowly. See? Nothing. Very slowly. I will try to tap to remove this air bubble, and then it is ready to use under the microscope. See, I will just tap it. No air bubble. And this is cheek cells and onion cells ready to view under the microscope. Now, to prepare fungus sample, this is Rhizobus. It is a black mold fungus, bread black mold. Remember, if you ever actually experienced black mold on top of the bread, we removed that bread and we add it in conservative. This is alcohol. Now, why do we add it in alcohol? We call it fixation in order to make the sample harmless. So this is why we add it in alcohol, let's say. It's a fixation. Fixation, we're adding the sample in alcohol to make the sample or the specimen harmless in order to work with. So since this is liquid, I either add directly from here into this slide and add stain, or you can just use it without a stain. It's very simple and very clear as well. But I will do both samples. So again, no need to put a drop of water. I will add very small drop of stain, because it's going to be a bit messy if I add too much liquid. I will try to mix what's inside the vial in order to have the sample, and I will take very small drop over here, and I will drop one drop over here with the stain. I'm gonna give it a little bit mix, and I will add my cover slip, but this is large. Oops, it's very large. You need to wear your size, but this is the one I can find right now. I'm careful, of course, because this is real fungus that I'm using. See, that's it. This is a fungus sample ready to view under the microscope. Very simple wet mount slide preparation, very simple experiment. It's a slide making to view cells under the microscope, and it's temporary slide. If I need to study that slide later on, all what I have to do is I will add glue to adhere the cover slip on top of it, and then I will call it a permanent slide. That's it. Okay.